Good afternoon. Thank you very much for coming. I'm a chair of the next session, so I have a pleasure to introduce myself. My name is Drazen Raucher. I got PhD in molecular biophysics, and after that I did a postdoc at Duke University doing laser optical tweezers, and also I did some biomedical engineering studies. At that time I was introduced in this elastin-like biopolymers, and since 2002, I'm a program director of our graduate program and professor of biochemistry at the University of Mississippi Medical Center. So today I'm going to talk about thermally targeted delivery of anti-cancer therapeutic peptides and also small molecule drugs using this elastin-like biopolymers, which is a really nice system. I'm going to explain you everything about that. First, just a quick overview about the current treatment of localized tumors. And then I'll just talk about background of ELP system, and later we're going to talk application of ELP system in cancer drug delivery, and I'm going to introduce two examples. One of them is going to be focused on targeting c-MYC oncogene in breast cancer and also in brain cancer, and then we'll see how ELP can be used for delivery of small molecule anti-cancer agents. So first of all, it's localized tumors. Basically, current treatment, it's a surgical resection, it's followed by chemo and radiotherapy. Usually that's limited by normal tissue tolerance, and also there's a chemo resistance and radio resistance. The main thing is with conventional chemo is that only a small fraction of drug reaches the tumor target. The rest of it's distributed through the body, and about only a few % is coming to the tumor, which is actually trying to reduce and shrink the tumor. So our goal, long-term goal, is basically to make this chemotherapy more specific and less toxic. So we are trying to use this system to deliver drugs specifically to the tumor site, and now I'm going to show you how is that done. Well, currently there are two ways to target drug delivery systems. One, it's passive targeting. Basically, passive targeting relies on this EPR effect, or enhanced permeability effect of the tumor. Tumor vasculature itself is different than normal tissue. It has a poor lymphatic drainage, and vasculature is different. So if you have a macromolecule, it's going to end up in a tumor just spontaneously. Well, in addition to that, there's active targeting, which adds special mechanism. It's like a special kind of missiles combination of ligands or something what specific for tumor. So you can add this to a macromolecule and can further actively target it to a tumor. In our case, we are interested in targeting tumors with a different mechanism, which is external stimuli, which is heat. I'm going to explain you how is that done. The base for this, it's this elastin-like polypeptide. Elastin-like polypeptides has been known since 80s, and it's a synthetic protein which consists of these 5 amino acids, VPG, XG, X could be any amino acids. It's thermally responsive, it's biologically inert, which is good, and it's easily expressed and purified from E. coli, and it can be fused to therapeutic peptides, or it can be conjugated to different small molecule drugs. So this is our elastin-like peptides. Below its transition temperature, it's extended and it's soluble in a solution. Once when you heat solution above the transition temperature, what happens is these hydrophobic sites inside are exposed, and then you have a accumulation or aggregation of the peptide. You can monitor this by turbidity. So below the transition temperature solution, it's really clear. After you heat solution, then you can see that turbidity goes up, and this is concentration dependent. So if you have a higher concentration of polypeptide, then concentration is lower. So you can finally tune this transition temperature to a tumor targeting range. Another very interesting property for this peptide is it can be easily purified. So it's expressed in E. coli. So what you do, you sonicate E. coli, you break down, you fractionate, you get a soup from E. coli. Soup consists of this basically brown parts, which are impurities, and white, it's our protein. So by increasing the temperature above transition temperature, protein, it's going to aggregate, and then you do a hot spin, you warm centrifuge, you get all your protein at the bottom of the tube, then you discard the soup, you have a nice clean protein at the end, and then what you do, it's fully reversible, so you can cool it down and all this high molecular weight junk, basically, it's going to be in the soup again. And again, what you do, you centrifuge in cold, so you can now remove soup and save, and you can do this several cycles. And that's what can be done several times. And as you go along, molecular weight is about 55, 56 kD. As you go along, after 3 or 4 cycles, you get a really nice pure band, pure protein that can be further purified with some other processes. So idea is, well, if you have this protein, you can attach either drug or you can modify it so it can carry therapeutic peptide. And then idea is you heat only tumor site, and next you inject basically polypeptide carrying a drug. So when it passes through the tumor, it's going to make aggregates. It's going to unfold and it's going to accumulate, and it's going to make like gel-like structure and release the drug slowly. After you stop applying the drug, basically everything is going to be cleared and the drug is going to stay in the tumor. Okay, so just to summarize, so what's the advantages of these polymer carriers? One, it's passive targeting. It relies on this enhanced permeability and retention effect. Also, it increases solubility and plasma half-life. I'll show you a few examples of that. And then that results basically in reduction of systemic toxicity. Further advantages of our system, ELP, it is polymeric carrier, but you can actively target it. It's thermally responsive, so you can heat just tumor site and get most of the drugs in the tumor. Most of all, it's genetically engineered, so it can be easily purified. You can manipulate the sequence, you can add anything what you want, and you can express in equally purified. So when I started this thing, our hypothesis is basically that systemically injected ELP will accumulate in locally heated regions, but it will circulate and eventually be cleared from the body. And I'm going to show this now for 2 examples of peptides and 1 example for small molecule drugs. So what's the ideal temperature for thermal targeting? Well, it's about 41 degrees, which is a high fever. So normal body temperature is 37 degrees. If you go more than 41, like 42, 43, then you get get additional problems like necrosis, maybe burns, tissue damage, things like that. So we are using here macromolecular carriers. They have increased solubility, increased plasma half-life, and also a high drug loading capacity. So you can load not 1, not 2, you can load many drugs on this system. And finally, hyperthermia is already used in clinics. So people realized that when they heat tumors, they have an increased sensitivity to chemo and to radiation. And also, you are increasing the blood flow to tumor, so what you do, you increase macromolecular extravasation. So just by heating and applying conventional chemo, you're going to get much better effect on tumor killing. So usually at this point of talk, people are already thinking, so how do you heat, right? Can you just heat superficially tumors? And no, basically today, at least in the United States, there are 200 hospitals which have this system. It's basically high-intensity focused ultrasound. So with a set of antennas, you have a high-convergent ultrasound beam, which is generated, and it can be basically targeted and seated to a deeply seated tumor. And also that can be monitored by MRI. So you can localize the tumor with MRI. You can heat it. And also there's some contrast dyes, so you can exactly apply a precise temperature for heat. So first, our attempt was to basically target therapeutic peptides. So therapeutic peptides, what's the advantage? Advantages, you use rational strategies. You can look at the crystal structure between 2 oncogenes and see how they interact, and then design peptides, so it's going to inhibit specifically that interaction. So they're highly specific for their targets. Disadvantage, they have a very short plasma half-life. They are easily degraded. They're substrate to many enzymes which are present in the blood. And they don't penetrate biological membranes. They can be also immunogenic. So taking advantage of our system, we are trying to overcome disadvantages of inherent peptides, which is, we are using now ELP. Well, ELP, it's a 55 kilodalton molecule which cannot obviously pass the plasma membrane. So in order to overcome this, so it can pass plasma membrane tissues, we introduce here cell-penetrating peptide. This is usually a small sequence between like 5 and 20 amino acids. It's usually charged, sometimes it's hydrophobic. And we tested many of them, like 10, 15, and these 4 are the best candidates which enable this construct to get into the cells and also gets through the blood-brain barrier, which I'm going to show in a minute. So that's one part which enables this to get in. And now we have this therapeutic part, which could be a drug. You can conjugate any kind of hydrophobic drug with proper chemistry. And it could be a therapeutic peptide. Therapeutic peptide, it's probably easier because this is genetically encoded. So you just have a genetic code of ELP. You modify this, adding amino acids which you want. You can purify, and then you can treat your cells and eventually animals. This is just a summary of peptides and some drugs which we delivered. I'm just going to explain today just a few examples. So what we have here, it's the first one is c-Myc inhibitor, which is overexpressed in cancer cells. And then my backup laser pointer. Okay, then you have a cell cycle inhibitor, which is a P21 inhibitor. Then you have NF-kappa B inhibitor, and there's some other inhibitor. For example, these 2 peptides inducing apoptosis. Also, with a proper modification of a conventional chemo like doxorubicin, Taxol, and also methotrexate, which are modified by adding hydrazine linker and also adding maleimide. This has been done with Dr. Kratz, who had a talk yesterday about albumin. You'll see some examples how this actually works. You can basically attach it to ELP and deliver it successfully in a model. So the first example is inhibitory peptide which inhibits c-Myc. So c-Myc is usually overexpressed in brain and breast cancer, and it's responsible for all this transcriptional activity which are basically pushing progression of the cell cycle and increasing proliferation of cancer cells. So what we did here is we designed a peptide which is actually inhibiting interaction between c-Myc and MAX. By inhibiting this interaction, you're stopping the flow of information here, and cell will basically get into arrest and undergo apoptosis and die. So this was done in vivo first, and then later on our ultimate target is to do it in vivo and eventually try to get to patients, but that's a long way to go. Let me just explain where we are. The first example is breast cancer model in mice. This is in E0771 cells. It's in a Jake model. It's a mouse breast cancer. It's very aggressive, and it will invade the peritoneal cavity and metastasize to the lung if it's not treated. Okay, so next, let's just see, can we localize this tumor? So we labeled our polypeptide with Alexa. BAC, that's one of the cell-penetrating peptides that can penetrate these tumors. ELP1, it's a thermally responsive polypeptide. We also have ELP2, which is non-responsive, so transition temperature is about 70 degrees. That's our control. But if you look at the mice, this is just saline. We didn't inject the peptide yet, so there is almost no background. Then if you just inject the peptide, there's no tumor. You can see that peptide will accumulate basically in the kidneys and also in the lungs. If you have a tumor implanted and you inject the peptide, there's some accumulation in tumor itself, and that's coming because of the EPR effect. The drug is going to get stuck in tumor because of the tumor vasculature. And finally, if you hit this tumor, you're going to get much more in. So we did quantify that in time, and as you can see here, it's after injection. So this is a non-heated, this is heated. There's at least 2-fold higher uptake when you heat the tumor, and eventually that's going to be cleared a few hours later. All right, so the final test is, let's go and see reduction. So these here are controls, basically saline and hyperthermia, which doesn't have much effect. And then you have BAC ELP, there's a slight effect, which is accumulation in the tumor due to EPR effect. And finally, when you heat, you have a nice tumor reduction. So here we have our control, which is ELP2. It's non-thermally responsive control. Also has effect due to this spontaneous accumulation, but as we already proven here, that can be really enhanced by using heat. Okay, so the next model, it's more challenging. It's a brain cancer because here you have a blood-brain barrier, which is impermeable for most of the current drugs. So for that, we use C6 red glioma. It's derived basically from methylnitrosurea-induced glioma in Wistar SARS. It basically resembles a human glioblastoma in histology. and also in rapid proliferation and intracranial dissemination. So this is kind of MRI image of the lesion. You can see this white thing here, that's the lesion. So we have several group of rats, and interestingly, without even heat, what happens is, so our polypeptide here is labeled with rhodamine. This is just ELP, no cell-penetrating peptide. There is some accumulation in the brain, but mainly in a ventricular cavity. And then if you have SIMB1, this is one penetrating peptide, there's accumulation in a tumor. And also with the other one, there's accumulation in a tumor and also in a interstitial space in the brain. So the second panel here, it's a dextran. It's a high molecular weight dextran, and that basically delineates where the blood vessels are. So the dextran stays in the blood, and if you do a merge, you can see there's some accumulation in a tumor here, but most of the accumulation here, it's really highly enhanced by introducing this CPP. So spontaneous accumulation in tumor even without heat, that's one of the characteristics of this polypeptide. So let's just quantify this. Saline, there is no signal. That's the ELP1 unheated, slight signal. Heated, there's a huge signal here. And that's our control. Again, there is a spontaneous accumulation. I showed it on a previous slide. With a heated, there is slight more accumulation, but not so much. So the panel, the figure B, it tells you there's like a threefold higher accumulation with a heat. Our control, there is a twofold higher accumulation, but that's not really statistically significant. We also look at distribution in organs. There is no difference heating a tumor or no heating, and this is our tumor here in the corner. This is already data which is shown in a previous panel. So the bottom line here, once when you inject and treat these animals several times during the several weeks, what you get is saline, and saline and hyperthermia and also BACH1, they have a moderate effect. They have a no effect, and they have a moderate effect on tumor reduction. But if you do heat these tumors, then you get a nice reduction of tumor growth. That's also reflected in survival. So basically, the longest survival you get when you have this CPP, LPH1, and heat. The rest of them basically dying much faster. All right, so the next application is, we did it with many other peptides, but these are two in vivo experiments which most convincing. The first thing what we did with small molecules is DOX. DOX is commonly used. We heard a lot about DOX in the last couple of days. So it's commonly used for a range of cancers, and it integrates into DNA. And basically, adverse effect of DOX, acute, there are nausea, vomiting, and heart arrhythmia. But the biggest problem is basically adverse effect of heart damage or myocardial basically dysfunction. So in order to get this DOX conjugated to ELP, we put on ELP cysteine. So the cysteine, and then doxorubicin was modified by Dr. Kratz by introducing this hydrazine linker and also maleimide. So this maleimide has a high affinity for cysteines. And next, we basically just introduced one cysteine at the end of ELP, and we got this DOX here. So our idea is, well, this DOX labeled ELP, once when we increase the temperature of the tumor, it's going to make aggregates, it's going to accumulate, have increased accumulation in the tumor. Okay, so why just one, you may ask? Well, we added two more just in case, so we can increase the drug load. If you go more than 3, then you you get change in transition temperatures. You may get micelles, so it's kind of getting complicated. So we found out that 3 is optimal number, so you have a high efficiency drug delivery. So first of all, if you just look at the free DOX, this is time scale, it's in minutes. So this is a plasma clearance, it goes out in 8 minutes. So this is really characteristic of all hydrophobic drugs. Basically, tumor gets only a fraction of the drug dose. If you attach this free DOX to ELP, so this is not heated and this is heated, you can see now we have this in hours. That's in minutes. So in 360 minutes, it still stays in a plasma. There is a slight difference between heated and non-heated. So the difference here is coming probably because of tumor accumulation, because when tumor is heated, most of the drug would be drawn from plasma and then end up in a tumor. If you look at the real concentration of the drug in a tumor, when it's delivered with ELP, that increases twofold uptake. With the dogs, it's about the same, so there's no increase in the dogs accumulation just by heating it. One of the problems which I just mentioned is accumulation in the heart. Well, in case of delivery with the ELP, there are no detectable level either with heat or without heat in heart. With the free dogs, after several hours, it basically there is some accumulation. So finally, tumor reduction. We have here control saline without hyperthermia, which doubles really quickly. Hyperthermia itself causes a little bit shrinkage of the tumor. And then we have dogs. So these two are dogs. And dogs and with or without hyperthermia reduces tumor somewhat. However, if you use our construct, you get a nice, almost complete reduction of the tumor. So we are not limited ourselves to dogs, which I just showed, but we also did some study with methotrexate, and also we did show in animals that you can deliver Taxol. We also got a nice reduction that was published in 2012. So in summary, what we do, we make these ELPs. We're doing simple molecular biology, then we purify it. We can purify it in high quantities, and then we do a tissue culture, checking for the activity, checking for the targets. We also do mice, but our ultimate target is basically to go to a human treatment. So I just realized that this is not a very good frame for human treatment. It should be a different color rather than black. It seems that this person is already dead. Huh? Okay, so I'm going to change this. So I want to thank my lab members, current lab members, and also I want to thank Dr. Bidwell and Dr. Mokhtar, who did most of the work. Our collaborators, these collaborators are mainly involved in a tumor reduction study in the brain, and Dr. Kratz, who actually synthesized all these chemicals. Funding came from NIH, NSF, DoD, and Vanderbilt Waste Foundation. And just a few things about future studies and collaborations. Basically, we are trying to deliver different chemotherapeutic. We have different targets like RAS signaling cascade, targeting cancer stem cells. And also now we are basically trying to apply ELP in treatment of spinocerebellar ataxia. We already published a paper about that. Delivery of therapeutic to cerebellum and also to neurodegenerative diseases. Gene delivery also, and we are working to making these biocompatible gels for wound healing and slow release of drugs. So if any of you, this is a platform what we have, and it can be modified. So if any of you is interested in collaboration or trying to deliver any kind of your drugs or systems, please feel free to talk to me or send me email after that. Before we leave, I just want to show you a short movie. It's only 1 minute and 15 seconds. This is just going to illustrate what I talked about. This is for non-science majors. Okay. So usually when I go and talk to people, okay, so this is, I have to go from the beginning. Okay, you see? All right, so this is, that's the polypeptides. It moves nicely. Okay, so in a solution, it's soluble. And when you increase the temperature, this is what's going to happen. It basically aggregates, it forms aggregates. So all these hydrophobic sites are exposed and it makes aggregates. So what we did in order to penetrate plasma membrane, we modified this peptide with cell-penetrating peptide, and then we added therapeutic peptide, or it could be a drug. The purpose for this exchange is to illustrate that this is like a Lego game playing blocks. You have a blocks, you can change this anyhow you want. For different tissues, you can use different CPPs. For different targets, you can use different therapeutic drug. So idea is you use one of these peptides and then you inject systemically, and now you have a heat. So heat is applied, and if you look inside the tumor, So these are some blood cells flowing. And then around the corner it's coming ELP carrying the drug. Okay, so increasing the temperature, you're forming this aggregate because you have a CPP that basically penetrates tissues and blood vessels, and finally tumor is going to become dead. That's the happy end. Thank you very much for your attention, and I'll be happy to answer any questions.
Thank you very much. Perhaps one or two questions. Well, if not, then I have one.
Yes.
No, he has one.
Thank you very much for a very excellent presentation and the nice video film you presented. How well this is extended in the segregation transition of the elastin molecules.
How sharp is the transition?
How sharp?
Okay.
If it is, if you can repeat it, and every time you get the same transition.
It's very
because it looks to be a very narrow.
It is
window.
very narrow. So the thing is, that's another advantage over this synthesized polymers, because there is no polydispersity. Basically, it's genetically modified, it's genetically engineered. So you always have a 55 kilodalton polypeptide, and it's always reproducible. Transition 1 to 2 degrees, and it can be finely tuned by changing the sequence and by changing the size of the polypeptide. So you can put it on, for example, if you want to make a hydrogel, so you can put it on like 33 degrees Celsius. So once when you apply it to skin, it's going to aggregate, it's going to form like a film. If you want something higher, if you want to stay in circulation longer without heat, then you can modify it to like 50 or 60 degrees. So it's very well defined. So that's another advantage of that.
The quick question I had is, you said you inject the stuff into the body, and then you heat up the tumor, and you said most of the drug ends up in the tumor.
That's the idea, yes.
And
in theory, yes.
Oh, okay, because when you said a twofold increased uptake, so my question is, what's the percent accumulation of the accumulation in percent of a total injected dose?
A percent, we didn't look this.
But that's the crucial number, actually.
Okay, so that's twofold. We are talking about a couple hours, okay? So what's really happening later, we don't know.
No. How much gets in, you inject a drug, and
Yes.
a certain percentage of a total injected dose has to end up in the tumor. And that's the important parameter. Doxil 8 to 10% as a standard. So what percentage of your drug ends up in the tumor?
Percentage is about 20 to 25% after 2 hours. So rest after that we don't heat anymore because concentration.
That's a lot. Very good.
Yeah. Concentration of the drug is getting lower as it gets clear.
Sure.
Because you have to have a certain concentration in blood plasma in order to induce this aggregation. So that can be repeated later.
Yeah.
You know, so the rest of it is probably going to end up later on because of the EPR.
25%, it's a lot. I mean, that's congratulations. That's great.
Yeah, yeah. Thank you.
Okay. Thank you. Any more? We have to continue now. Okay. Thank you very much again.
Thank you.
And you get also a certificate. Please. No picture taken.